Flow cytometry is a powerful tool used in cell biology and medical research to analyze and characterize individual cells within a heterogeneous population. It allows researchers to measure numerous properties of cells, such as size, complexity, and the expression of specific proteins or molecules. This technology has revolutionized the field of immunophenotyping and has become an indispensable tool for investigating diseases, studying the immune system, and monitoring treatment efficacy. In this script, we will delve into the various aspects of flow cytometry, including its principles, instrumentation, data analysis, and advanced applications. So let's begin our exploration of flow cytometry. Flow cytometry is a technique that combines principles from physics, optics, and immunology to analyze cells or particles in a liquid suspension. It allows researchers to examine and quantify individual cells based on their physical and chemical properties. The main components of a flow cytometer include a fluidic system, a light source, usually a laser, and detectors to measure the signals generated by the interaction between the cells and the light. By analyzing multiple parameters simultaneously, flow cytometry provides detailed information about different cell populations within a sample. Fluorochromes are essential components of flow cytometry as they enable the detection and identification of specific biomarkers or proteins on the cell surface or within the cell. These fluorochromes are conjugated to antibodies, which target specific proteins or molecules of interest. Antibodies play a crucial role in flow cytometry as they recognize and bind to specific epitopes, allowing the identification and quantification of different cell populations. The choice of fluorochromes and antibodies depends on factors such as the desired targets, spectral overlap, and instrument capabilities. Flow cytometers consist of complex instrumentation to analyze cells efficiently. The core of the instrument is the flow cell, where the cells pass through a narrow stream. The flow cell ensures that the cells pass through the laser's focal point, allowing efficient interaction between the cells and the light. Different types of detectors are used to measure the scattered or emitted light signals, including forward scatter, FSC, side scatter, SSC, and multiple channels for fluorescence detection. The optical configuration and filter setup are critical to ensure minimal spectral overlap and maximize sensitivity. In flow cytometry, compensation is necessary to correct for spectral overlap between different fluorochromes. This correction is achieved by using compensation controls, which consist of single-stain samples where each fluorochrome is separately labeled. These controls help align the spillover signals detected by different detectors, ensuring accurate interpretation of the results. Proper compensation is crucial for robust and reliable data analysis, avoiding misleading conclusions due to spillover artifacts. Flow cytometry generates vast amounts of data, requiring specialized software for analysis. Data analysis involves analyzing histograms, dot plots, and other graphical representations, enabling the identification and characterization of specific cell populations. Common analysis techniques include gating, which defines regions for specific cell populations, and statistical analysis to quantify data. Advanced analysis methods, such as clustering algorithms and dimensionality reduction techniques, allow the exploration of more complex datasets. Immunophenotyping is a widely used application of flow cytometry that enables the identification and characterization of different immune cell subsets based on their surface markers. By using specific antibody panels targeting a combination of markers, researchers can classify and quantify various immune cell populations in a sample. Immunophenotyping has significant diagnostic implications, providing valuable information for immunodeficiency disorders, hematological malignancies, and monitoring responses to immunotherapies. Flow cytometry also allows the analysis of the cell cycle, providing information about the distribution of cells in different phases of the cell cycle. By staining DNA with fluorochromes, researchers can distinguish cells in G0/G1, S, and G2/M phases, as well as detect aneuploid populations. Cell cycle analysis is crucial in studying cell proliferation, evaluating cell cycle regulators, and investigating the effects of drugs or treatment modalities on cell division. Flow cytometry extends its capabilities beyond surface marker analysis through intracellular staining. This technique allows the detection and quantification of intracellular proteins or cytokines of interest. By permeabilizing the cell membrane and utilizing specific antibodies or fluorochromes, researchers can analyze intracellular signaling pathways, measure cytokine production, and investigate cellular responses to stimuli. Flow cytometry is capable of detecting and analyzing rare cell populations within a sample. This is particularly useful in fields such as cancer research, where the detection of minimal residual disease or rare circulating tumor cells is crucial for prognosis and treatment monitoring. Methods such as enrichment techniques and multi-parameter analysis help identify and characterize these rare events, enhancing our understanding of disease progression and therapeutic responses. Flow cytometry sorting allows researchers to isolate and purify specific cell populations based on their characteristics. This technique is valuable for downstream application. This technique is valuable